So hello everyone. Thank you all guys for this great opportunity to share our recent work on the mRNA vaccine elicited antibodies against the SARS-CoV-2 and circulating variants. And as introduced, I'm Zijun Wang from Nussenzweig Lab at the Rockefeller University. So according to most updated, SARS-CoV-2 has infected more than 120 million individuals, which results in over 2.65 million deaths worldwide. And multiple SARS-CoV-2 variants are circulating globally. Several new variants emerged in the late of 2020, most notably the UK variants, South African variants, and the P1 variants that in Brazil. The E484K, which is the spike protein mutation, affect the neutralization by both polyclonal and monoclonal antibodies. And we know that the newly identified UK variant is now carrying this mutation. Even though it is still unclear about the effects of these variants, these mutations on disease severity, outcomes, and death rates, many studies suggested that this mutation may affect the transmissibility and antigenic profile, which may affect the ability of antibodies generated through natural infection or vaccination to recognize and to neutralize the virus. And there are other two variants that are circulating in the US now. One is the California one, and the other one is the New York variant, which was recently identified and carries the E484K mutation also. Unfortunately, most of the recent data, including ours, finding that the variants of concerns may evade our protective neutralizing responses induced by pre-oral infection and or by vaccination, particularly after a single dose. Thus emphasize the need to generate highly potent immune responses through vaccination to thus provide us protection against the emerging variants. And many vaccines are developed in the past year, and most of them are not in clinical trial stage. And these vaccines are designed with different technologies, including the inactivated vaccines, where the immune response is elicited. It's directed not only against the spike protein, but also against many other many other SARS-CoV-2 antigens. And another one is the based on the protein unit, which is a large trimeric aggregate of the spike protein that protrude outside the virion, which is the essential role in the docking of the SARS-CoV-2 to the human cells. And there are so many vaccine projects based on the SARS-CoV-2 proteins or their fragments. And the Novavax is one of the examples where the recent announced data is quite inspiring. And other vaccines, for example, the one that based on the viral vectors, including the Johnson & Johnson and the Oxford AstraZeneca one, which this seems to have some problems recently. And for us, I guess the vaccine that we care the most is the mRNA vaccine, which most of the US population already got or will get soon, including the Moderna mRNA 1273 and the Pfizer-BioNTech BNT162b2. Here's a summary of some top 6 vaccines created by Crystal. As reported, the efficacy of all these top 6 are above 50% against the SARS-CoV-2, but the efficacy against the variants, especially against the South African one, dropped a lot. For example, the Australian one dropped to only 10% against the South African variants. And this is consistent with many recent studies show that the neutralizing antibodies elicited from vaccinees were as effective against the UK variants, but only about 1/5 to 1/10 as effective at neutralizing the South African one. So our question here is, how do the mRNA vaccine elicited antibodies respond to SARS-CoV-2 and circulating variants at molecular level? First, this study included participants recruited between last October and this January 2021. And 20 volunteers were involved in this study, which received 2 doses of the Moderna one or the Pfizer-BioNTech mRNA vaccines. The median age of the volunteer is 43, and the time from the second vaccination to sample collection time varied between 3 to 14 weeks, with an average of 8 weeks. And none of these volunteers had a history of pre-oral infection or serious adverse events after vaccination. So first, we measure the plasma, IgG, IgM, and IgA responses to SARS-CoV-2 spike and to the RBD. And all individuals tested showed reactivity to spike and RBD and was significantly higher compared to the previous COVID historic controls. And both the anti-spike and anti-RBD IgG levels were higher than that measured in the IgM and IgA. And there we found a very strong positive correlation between the anti-RBD and anti-spike responses in all the 3 immunoglobulin isotypes measured. And also similar to previous studies that the IgG and IgM levels were significantly higher in the vaccinated group compared to our previous cohort in the convalescent group that assayed 1.3 months post-infection or 6.2 months post-infection, while the IgA levels were similar. And plasma neutralizing activity was determined using the pseudotyped SARS-CoV-2 virus. And consistent with some previous studies, there was a variable range of plasma neutralizing activity 8 weeks after the vaccination, which was similar to that elicited by the natural infection in our previous convalescent cohort assayed after 1.3 months, but greater than the activity at the 6.2 months. And we found there was no significant difference in neutralizing activity between the Moderna and Pfizer-BioNTech vaccines. And when we do the analysis, we found it interesting that the RBD binding and the neutralizing activities were directly correlated with the time between the first time of the vaccine dose and the blood donation, with significantly reduced level with time in both cases. And then we tested whether the plasma from vaccinated individuals can neutralize the pseudovirus that carry the mutations in circulating SARS-CoV-2 variants of concern. We saw that the vaccinees were significantly less effective in neutralizing the pseudotype SARS-CoV-2 mutants. We also tested our convalescent plasma obtained at 1.3 months and 6.2 months after infection, and we found that they were less effective in neutralizing the K417N, E484K, and the N501Y combination, which we call it CAN. Well, what we learned from here is that the plasma neutralizing activity elicited by either mRNA vaccine or by the natural infection through natural infection, although variably, but significantly less effective against the pseudotype virus experiments, the pseudotype virus that carry the RBD mutations found in the circulating emerging SARS-CoV-2 variants. And we then used the flow cytometry to analyze the circulating SARS-CoV-2 RBD specific memory B cells that elicited by the mRNA immunization. We can see from here that the percentage of the RBD binding memory B cells in vaccinees was significantly greater here, greater than the naturally infected individuals assayed after 1.3 months, but similar to that same individuals assayed after 6.2 months after infection. And mRNA vaccination elicit robust SARS-CoV-2 RBD specific B cell responses that we can see from here that resembles the natural infection. And next, after the BCR sequencing, we obtained around 1,400 paired antibodies, heavy and light chains from 14 vaccinated individuals. And this pie chart here shows the clonality of the antibodies that isolated from each individuals. And the color slice indicate expanded B cell clones, and the white one indicate the singulates. And the black outliers indicate the percentage of the clonality. And we found here that the expanded clones comprised like from 4% to 50% of the overall RBD binding B cell compartment, and the clonality is not different between the natural infection and the vaccination. And we also examined the V gene usage in these vaccination-induced antibodies. And we saw that it is very similar to what we observed in natural infection antibodies, the IgVH353, VH330, and the light chain genes, the V-kappa 139, 133, and also the lambda V gene 321 are overrepresented in the RBD binding memory B cell compartment of the vaccinated individuals as compared with the human B cell database. And another interesting thing is that we found that when we analyzed the antibodies that share the same combination of the IgVH and IgVL genes, we found that these antibodies from vaccinees comprised 39% of the clonal sequences. So here, we marked the individuals that received the Pfizer vaccines in red, and the black are the Moderna ones. And each slide here showed a clone, and the white are the singlet antibodies. And we marked here in green that links are clone in this patient and in these individuals, and a clone, and the green link two clones, and also the purple links a clone from this individual and a singlet in that individual, and also the gray links to the gray line. The gray lines links to singlet. And more strikingly, we found that the overlapping sequences become 59% when combined with the natural infected individuals from our previously cohort. And here are the two examples showing that some nearly identical antibody sequences in both showing the heavy chain CDR3 amino acid and the light chain CDR3. And we can see that these start showing that identical amino acid and very few changes we found like in the antibody sequences in the individuals that received either Moderna or the Pfizer vaccines. We also checked the number of V gene somatic happy mutations. We saw that the antibodies from the vaccinees have higher number of somatic happy mutations than in natural infected individuals as at 1.3 months, but lower than that in the same individuals as at after 6.2 months in both heavy and light chains. And we think this may largely due to the sample collection time after vaccination, which indicates that the germinal center really needs time for it to accumulate certain number of somatic happy mutations in the case of SARS-CoV-2. And altogether, we expressed 127 representative antibodies from these vaccinees, and we tested them. The binding activity of the antibody elicited from vaccinees we found was higher than that observed in infected individuals after 6.2 months, but similar to that obtained 1.3 months after infection. We then tested the binding of 17 selected monoclonal antibodies from vaccinees to RBD mutations. And we found that 1/3 of the antibodies tested showed at least more than 5-fold decrease the binding to at least one of the mutant RBDs as shown here. And we can see that this group of the class 2 antibodies totally lost their binding to the RBD when there is E4A4K mutation. And on the other hand, which is consistent with the plasma neutralization results, the IC50 of the vaccine antibodies, monoclonal antibodies, were not significantly different from antibodies obtained from naturally infected individuals after 1.3 months or 6.2 months. And in a previous paper from Christopher Albanese from Pamela Bjerkman's lab at Caltech, where they successfully classified the antibodies that were isolated from the convalescent individuals by using the cryo-EM, depending on how the binding pattern in the RBD, they concluded that there are at least classes of neutralizing monoclonal antibodies regarding to the RBD. And in this current study, Christopher and Zhi from Pomelos Lab did the same thing for the antibodies to see how they look like. Here, what they found is that the majority of the monoclonal antibodies from the vaccinees can be characterized to recognize the epitopes that included RBD residues involved in ACE2 recognition, which is exactly what ours and others found in natural infected elicited antibodies. That also the antibodies classification is very similar to what we found before. And we concluded here that the RBDs of the spike trimer translated from the mRNA vaccine adopt similar conformation as observed on the structures of a trimer that on the surface of the SARS-CoV-2 virus. We then checked the neutralizing brief of the monoclonal antibodies and mapped their target epitopes. We tested the several number of most potent antibodies against a panel of 12 SARS-CoV-2 variants, and most of them represent circulating variants and have been associated with rapidly increased case numbers. Here we found that the K417N is resistant to the class 1 antibodies, and here the E484K is resistant to our class 2 antibodies. And the N439K is resistant to class 3 antibodies. As seen in natural infection, we found that 9 out of 17 of the antibodies that we tested were at least 10-fold less effective against the E484K mutation. And in addition, five of the antibodies was less potent against the K417N and 4 against the N501Y by 10-fold or even more. And additionally, we want to know whether antibody-imposed selection pressure could drive the emergence of the resistant mutations in vitro. And this was done by Paul Binash's lab, where they cultured and recombinant VSV virus in the presence of each of the selected neutralizing monoclonal antibodies. And all of the tested antibodies selected certain RBD mutations. And notably, that 6 of the antibodies selected for this K417 mutation and 5 of the antibodies select for the E484 mutations, and we found 3 selected for the N501 mutation, which coincide with mutations that present under circulating variants. And similar results were obtained with antibodies being developed for clinical use, including 2 combinations isolated from our lab, the C144 and C135. We found that even though some certain mutations were resistant to an individual monoclonal antibody, but the antibody combination remained effective against all of the variants that we tested. That's further confirmed that it's really important to use the antibody combination in the clinic. And similar results were also found in this separate study from host lab, where they tested a number of therapeutic antibodies, including our C135 against the mutations in the UK or South African variants by using the real virus. Well, we can see that this antibody is really standing out. So our summary here is that the mRNA vaccine elicited nearly identical antibody responses against SARS-CoV-2, specifically the RBD to natural infection, including the fraction of the memory B-cell compartment and the B-cell repertoire and antibody classification based on the cryo-EM, and also the sensitivity to the emerging SARS-CoV-2 variants, and also the selection of very similar RBD mutations. And the other part is that these weakened antibody responses against circulating variants could or might be compensated by the antibody evolution, which we already showed in another study showing that evolution over time from 1.3 months and 6.2 months, and also the polyclonal nature of our serum. And another important thing is the T-cell immunity. And our conclusion here is that the mRNA vaccine and natural infection elicited similar and potent neutralizing antibodies and memory B-cell responses, and the SARS-CoV-2 vaccines and antibody therapies may need to be updated in order to compensate for the viral evolution. So many thanks to all our collaborators and also a great team at Rockefeller University, Michel Nussenzweig Lab and Combinationswere Lab, and also Marina Kasky, who leads our hospital program, and also our great collaborator, Pamela Bjorkman's lab at Caltech, and also Raphael from NIH. That's it. I'm happy to take questions.
Thank you very much, Zijun. That was a wonderful talk. I have a question. It's mostly a clarification. In any given patient, either convalescent patient or a vaccinee, you do generate all 4 classes of antibodies, correct?
No, actually we find like the class 1, 2, 3 antibodies, but we didn't see class 4 antibodies. But I mean, because we only get 17 antibodies that we want to dissolve the structure. So not all the antibodies that we do that. And also we also find some antibodies that not very specifically can be classified into class 1 or class 2 because they share some characteristics that either class 1 or class 2 or class 2 or class 3. And also another thing is that we identified a group of antibodies that was by our ELISA data, we mapped with different mutants, and we found it cannot be classified for the ones that identified before. But basically, it's very similar to what we found with the natural infection elicited antibodies, which can be classified to at least 3 groups.
Got it. And for the time being, there is no naturally occurring viral mutant that is effective against all those major classes of antibodies. It's either one or the other or the other. Is that correct?
As far as I know from our study, yeah.
Okay. We have a question from Rodney Rothstein. Rodney, you should be able to talk and unmute yourself.
Yeah. Thanks, Eugene. Very, very interesting talk. I have a question about following people after their first dose of the vaccine to the second dose of the vaccine, and then to 2 to 6 months out. Have you done any studies like that? Because that would fit into what you talked about at the beginning of your talk.
Yes, that's a very interesting question, and I think we will find it out.
I'm looking forward to those results. Thank you.
Yeah, thanks.
We have a question posted in the Q&A by Laura Landweber. I'm not sure if Laura wants to read this herself. If she does, please go ahead and raise your hand. There she is. You should be able to talk, Laura.
Hi, thanks. Yes, I'm very interested in the question, as are many of us interested in whether or not the data for new local individuals who do test positive if that can be coupled to whether or not they've been vaccinated or received the second dose.
Yeah, for now, I don't have like a very large population to say that, but actually we do see our people that get infected, but after the vaccination. So, and most likely that one gets infected with the New York variant because we identified here. So yeah.
So we can't.
But that is the only one, yeah, we saw before.
Mm-hmm. So we don't know what protectivity we're able to transfer with the vaccine.
Yeah, I think it's hard to say for now, but we will find it out.
Do we know what New York City's current rates are? Because I know they were climbing very rapidly. That is the current rates of the E484K mutant.
You're right, yeah.
The Washington Heights variant, for example. Is that, well above 50, 70% or anything like that, or do we not yet know?
No, I don't think we have any idea about that, but yeah.
All right, thank you. Well, I'll keep a close eye on it.
Yeah.